if you're a Windows user, if you did not get in something bash-like, then could you raise your hand, or the green and red stickies are if you need help. So the green and red stickies, can we pass them through again? Because I think more people have shown up, so just pass them backwards. Everybody needs a green and red sticky if they're taking the class. It's not red, it's pink, but it's supposed to be red. I couldn't find red Post-it notes. So you ended up with pink. If we want them. I'm going to do the first 3 sides. They're what we're doing. Unless you don't want to do any. Okay, go for it. Yeah, go ahead. Actually, let's sit next to Grant. Go feel more comfortable. So if you are a Windows user and don't have Bash installed, put a red sticky on your computer so one of us can come help you. Actually, I had them use, what was it called this year? It's on the website, if you go to the website real quick. I think it's called the Bash Windows user Ubuntu. It looks just like a normal terminal, which is really nice. And the directions are right on the website, and I've got troubleshooting notes if you guys need them. Actually, I should keybase that to you guys. Got 1 minute. Let me keybase the, is this the, yep. It is, yes. Oh, boy. Yeah, I know. Zach went upstairs to get board cleaner and markers. So at my office when I first went back, it is now indexed.html. Yes, you can just do shorthand slash and it should go through. And you should be able to read all of that. You don't have Post-its, everybody that's attending will need a green and a pink one. They should be going around. I will make the statement that the university policy regarding COVID is that masks are optional, but that we're supposed to be sitting at 6 feet intervals. It probably means every other chair. I'm not going to enforce it if you're comfortable sitting like whoever you're sitting by. But I am going to wear a mask when I come help you. But when the helpers come around, we're all vaccinated, the helpers, all of us. Just in case. But if we're sitting over your shoulder, we'll wear a mask. And for those of us in the lab that showed up that didn't bring a mask, there's a bunch of disposables here. Unfortunately, E-Times is not back yet open, so there's no coffee in the building, so everybody brought their own. It's impossible to code without coffee. She doesn't think so, but I do. I think so. My grad school advisor used to say that it was coffee in and code out. That's right. I agree. I appreciate the importance of coffee in and code. Thank you, Zach. No problem. Partly. I'm not going to wear a mask while I speak to you, though, because it's really hard to hear somebody in a mask. But if I come up behind you, I will be wearing one. Because there will be a lot of close contact and us over your shoulder, so. Pretty sure these haven't been washed since 2019. Yeah, we may very well be the first class in here since lockdown. Okay, last time I'll announce it. If you have a Windows PC and you did not get the Bash Ubuntu terminal installed, please put a red sticky note on your computer. If you need red and green sticky notes, raise your hand and we can come bring them to you. Okay, red and green sticky notes back there. Do we know where they are? Does anybody have them?
They're not red and green. They should be red and green. I couldn't find
Red
red.
and green, they're more like neon and pink.
That's what I found. It's close enough.
So no one else has problems with green notes? Should run red sticky on a piece of here.
Yep, she's working on it right now. We need some sticky notes up here, Lynn.
Graham? Sam? And Ariel? Ariel, Sam? Graham?
Oh, yeah, I think it's 1 to 4.
Okay, red and green sticky notes. Do you have any? Right there.
If you want to help somebody, guys? There's
Box of them.
We brought them on purpose.
If you're within 3 feet of somebody, you have to wear a mask. So people have to wear a mask when they go people over the shoulders. I'll just. No, it's only 3.
Okay, awesome. I must have been looking at the 29th.
Okay. Hi, everybody. Welcome to the short read workshop.
I'm Robin Dowell.
I'm Mary Allen.
We run 2 labs on the 4th floor of this building, doing lots and lots of genomics. The goal of this 2-week workshop is to bring you guys up to speed on using the high-performance compute systems. Some of them are available on campus, it's also available on Amazon. We'll be using an AWS instance for this class. This week is really about learning to use the computers, and next week is about learning to use those computers to do data analysis. It's not quite that cold and separated, it's like you get roughly the idea. I'm going to introduce all of the people that will be teaching this week, because it ain't me. And that will be helping you. The point of the 2 little colored Post-its is every time you run into trouble, you put the red one, pink one, on the back of your computer and somebody will look help. There will be times when we will say, okay, everybody get that, and if you did, we'll use the green one at that point to indicate what you did. So that's how we track the pace of the class. So let me introduce my lab. This is Zach, Taylor, walking down the aisle. This is Lynn, Ariel, Kendra's in the back. I'm missing some people. Chi, Jessica, I know Chi's here somewhere.
There he is.
These are the guys who will actually be teaching.
Sam's
this
up
week.
front. You missed Sam.
Sam's up front.
Do we miss anybody else? Oh, Gilson's hiding in the crowd, too.
There's Gilson. I know he's here earlier, but I can see that. So these are the guys who will actually be doing the teaching. They'll also be coming around and helping when you have the right stickies. Okay? I'm going to turn it over to Mary. She's got today.
Okay. Every day I'm going to start with a capstone that just tells us the point of each day and how it relates to the rest of what's going on. So, point of today is to introduce you to what's going on. Oh, now I have to figure out how to use these. There we go. Okay. Schedule for week 1. Like Robin said, week 1 is mostly using the computers. We'll be using Vim, Linux, supercomputers. The only thing that's really related to short read will be on day 4, which is mapping and quality. And then day 5 is a quiz. And we're doing a quiz because this week is learning how to drive and next week is actually doing the analysis. And if you can't drive, that's going to be hard. So on Friday, we give you a quiz, and depending on how fast you go through it, you know how much you have to practice over the weekend. Some of you will get that quiz done like this, and you don't have to practice over the weekend. If that quiz takes you longer than our time in class allows, you will practice over the weekend because next week we'll be going even faster. Okay, next week is RNA-seqs on days 6 and 7, nascent and single cell on day 8, ChIP-seq and ATAC-seq on day 4, or sorry, that should be 9, that's weird. And day 10 is what do I do after I get all that out of the analysis, okay? Come to them even if you're not going to be doing those things, because every time you practice using Linux, Unix, you will get better at it, and you will be able to do your analysis better even if you're not planning on doing ChIP-seq, ATAC-seq, or RNA-seq. Okay, today we're going to be doing capstone day 1, which is what this is, and then we're going to be logging in and out of the supercomputer. Not all of you will do that successfully. That's okay. The system is not always set up on the first day. That's why we start with logging in and out, because we need to find out who needs help to get themselves on the supercomputer. So it will be very quick in and out. Then Lynn will go over an overview of Illumina sequencing. We will start teaching you Vim Tutor. Vim is the program that is basically the Microsoft Word of Linux. It is a much steeper learning curve. In other words, it will take you a day or two to get used to this program. So we're going to practice it. And then we will also log on to the X2Go client, which is how you will visualize data on day 4. Like I said, the logging in today is mostly to get us to get you on the machine, because tomorrow we got to have you all on the machine. You will have homework. We will talk about your homework, but the homework will be day 1 and day 2 videos, because you probably haven't done day 1 videos. I didn't give them to you. Vim Tutor, and then if you get through Vim Tutor far enough, I want you to keep going, because if you get through all of the Vim Tutor, you will be much more comfortable for the rest of the week. So try and get as far as you can on Vim Tutor today. Okay, when you think about short-read sequencing, there are 2 ways of thinking about it. Most people think about it 2 ways. One is the path, like how do I get from your sequencing file, which is in the top, to your publication. And one of the things we're going to go over today is the path. But the basic path stays the same no matter what you're doing, RNA-seq, whatever, ChIP-seq. You map the reads, and then you do a bunch of counting and statistics, you look at your data, and you get your publication. There's your basic path. Some people would prefer to think of it as the stops on the path. So if you're going on a road trip where you're going to be stopping, and that's where our files types are. Yeah, it doesn't look good at all on this. It looked okay on my screen. But those are the file types. You can look at this more in the video that I gave you guys. This is what we're going to be learning this week. What is the path? What are the file types? Now, when you put it all together, it looks pretty complicated, but we're going to break it down into individual steps per day so it doesn't get too complicated, hopefully. What really matters in this class is you've got to learn how to drive. So all of you are used to using computers. Probably none of you are used to using Linux. Maybe some of you have done some Linux command line. So the point is, the hard part of learning sequencing is not learning the path. You can look that up. You can read that. You probably all have an education that allows you to read it. The hard part is learning how to drive. Because without somebody over your shoulder telling you what the mistake is and how to fix it, it's really hard. That was the part I struggled with because I learned Linux and Unix on my own, and I had no one sitting over my shoulder. So that's what this class is for. Someone will be around to sit over your shoulder and remind you to use your left blinker. And that's what your helpers are for. You have red and green sticky notes. I'm sorry, they're pink and green. I couldn't find red. But red means stop. I can't go any further without your help. So stick your red one on there when you need help.
Back in your computer, we can see it.
Yep, so you want it on the back of the computer right here, because then we come up here and we look and we can easily spot and help. The green one is when we're trying to assess if we're ready to move to the next step. So we will ask you, put your green sticky note if you are done with this step. And by telling how many people have green sticky notes, we know if we can move on or not. Okay, the other thing I want to remind you of, the mistake you will make most often is spelling and capitalization. So before you call somebody, and we've told you to do something, make sure your spelling and your capitalization are right, because that is the most common mistake. You will make this mistake. I still make this mistake. It's totally normal. And you're probably going to feel like this dude who's driving, and that's okay, because when you're learning Unix and Linux, that's how it feels. You're a little stressed. You're like, why isn't this working? It should be. That's normal. Okay. Not moving forward. Oh, yeah, you're right. That is the last thing I had to say. Now Lynn's going to take over and teach you guys stuff.
All right. So the first thing that we're going to do is we're going to log in. So this is directly from the PDF instructions that are on the website.
I was going to show them the website.
Okay.
Can you show them real quick?
Yeah, sure.
It is now, let's just go to DnA and link through there.
Okay.
So go to dna.colorado.edu to start it.
Okay. So we're going to go to education, and then under the short read sequencing workshop, online videos and materials, and that'll pull up the class page for the rest of the class.
So dna.colorado.edu, click on education, and then on the short read.
Or the URL is the far right front board.
Yep.
And that'll take you straight to that page.
So the top here is all the general information about the class. Hopefully, most of you were able to do all of the steps in before day 1, and then we're going to have a day, or a section for each day that has the videos for that day, any in-class materials that we've posted, as well as homeworks and other stuff that might be useful. Okay, so under day one here, there's a document in in-class slides that is logging into a supercomputer. It's also in the libraries and sequencing PowerPoint, which I am showing right here. If you want to reference that on your own machine. So, I don't know how many people tried this at home, but generally, these are the steps. I'll show you what it looks like on my machine, which is another reason why it's useful to have it on your machine, because during the class, we're going to go back and forth. People in the front are going to go back and forth between the slides and their own terminals to show you kind of a more in-motion demonstration. So it's useful to have these on your own computer in order to reference them. So really, once you can get a terminal open, which hopefully almost all of you can, then you just do SSH and then your identity or username at that IP address down there. Is it big enough for everybody to see? We can draw it bigger.
You can also just make your screen bigger and type it in.
That too.
And then it should be big enough for them to see.
This will be for the rest of the class, though.
So if you cannot open a terminal, type your username at that number and hit enter. Please give us a red sticky note so we can help.
Okay. That is the IP address for the AWS server that we will be using for this class.
So red sticky notes if you need help, please. And green sticky notes if you are logged in already. If you're on a Mac, you're looking for terminal. If you're on a PC and you followed our instructions, you're looking for button.
That should be it.
Yes. Oh, if it asks you for a fingerprint key, she's already logged in, so always type yes. Yes.
Yeah, you would say that.
It will only ask you yes or no the first time you log in. It just wants to know this is the same computer.
Just in case.
That's what we have. Okay.
Oh.
And we're in. Oh, is this? So yeah. So it is not your identity. It is the username you gave us for GitHub. GitHub username.
Yeah, I'm having trouble too.
Please give us a green sticky note if you're done. And for the people in the Dowell Allen Labs, please look under users for their username to make sure they're actually in our list. Good. First thing,
Is that it? Yeah, I'm not gonna.
okay. So for those of you who have permission denied, we need to set up a GitHub SSH.
It's almost here.
Did you say Mary? Or GitHub SSH key. It's most likely their SSH key that you set up. Okay, so let's set up an SSH key, Lynn. Lynn's going to try and walk through the group for those who are getting permission denied. She's going to try and walk through getting you an SSH key.
All right. I'm getting permission denied too, so.
Oh, are you?
I am.
You were in earlier this week though, right?
I did it last week, yeah, it was fine. Okay. Is that document not on the,
Oh, is it not?
it's not on the before day one?
I'll do it real quick.
Okay. I'll find it though.
I'll link it.
Oops. do. Extend.
Don't worry, this is normal, guys. So email Max, with SR. That IT is,
And just have that person email.
no, you can do it. I do it. Yeah. For the AWS. SSH, yeah, SSH, I'm getting it down right now.
All right. So, did everybody get their GitHub usernames added to the document?
If you do not give us your username by noon on Friday, Kendra's collecting a list, she's up there in the corner, you go give her your GitHub username. If you did give me your username, we're gonna go to SSH key setup. And do you want me to just keybase it you so it's faster?
I have it.
You have it? Okay.
It's up here.
And I'll put it in. Okay, so if you're a red sticky note, we're gonna go to key setup.
All right. Whoop. All right.
Okay, guys, first thing you do.
Ssh Keygen.
Yep, are you guys watching? Follow along in your terminal.
Are you showing how, or Lynn,
Yeah.
are you showing how to generate the SSH keys?
Say that again?
Are you going through how to generate keys?
Yeah.
Awesome, thank you.
Yep. So I'm really just following this line right here is the exact thing that I'm typing down here, except put in your own email address. You don't need a passphrase on this file. It will, once it's generated,
So just hit enter twice.
yep, just hit enter twice and it will generate a key. I'll go back to this command right here. Is everybody with me?
Anybody else not give their username to us on time? Heather's collecting them. No, aren't you in the middle of mine? Yeah. Then you did. Oh, okay. Your letters. SSH, I'll put this. I'm trying to find that file. I will put that file on the website real quick.
The config one, or this one?
That one. Trying to figure out which, I'll put it on Google Shares, but I've got free accounts.
All right, I already have this config file.
That document?
That one, yeah.
Okay. We're gonna have that document up on the website in a few minutes. If you
But if you don't have a config file under your home.ssh/config, then you can download the one from our website at that URL. Curl is a command that downloads. All right, if you're on a Mac for this SSH add, you use that -K flag. If you're not, then you don't need that flag, as far as I can tell. Say that again?
I'm working on it right now.
Yeah,
I'll be done in just a second.
yep, you will shortly.
As we speak.
All right. When you can either, if your Linux has a PB copy, you can PB copy. Technically, I shouldn't be showing you my private key, but I don't think anyone is going to be writing this down.
We're taking pictures.
Or taking pictures. But if you
You're
don't,
being recorded, but you can change it later, it's fine.
I mean, it's for AWS, right? So you can say cat, that file right there, and get your private key.
Yeah, Mary, we did that.
Got it, working on it.
And as this document says, you can just copy that private key into a section on your GitHub where it says SSH Keys. At the very top? Oh, there we go. Yep. Okay,
This document will also be up on the website in just a moment.
right there.
Right now. Oh, you can do that. You do that while I help her.
Also, the link seems to go somewhere.
Can you show the users list real quick? So on the machine.
I'm not on there.
Oh, okay. Yeah, that's right. Let me, you're on there? Is it letting you on?
Give me just a sec.
Okay, so the link is now on the website. However, when you load up the website, because it just changed, you have to hold shift when you reload that website or it will not show you the change. So the link is now present on the website. All right, show them the.
Still giving me a permission denied. I don't know what's going on.
You can always see.
Oh, well,
Why not?
to be fair. That might be a,
Even after
GitHub
key?
needs to reload. Could be.
Oh, do remember that once you generate the key, it takes 5 minutes. That could be the problem. No, once you generate the key, it takes 5 minutes. So if he did not generate a key on your computer, it would not be ready. Yeah? Sorry, I can't hear you. Oh, you changed the link name? There should be two that say library, Illumina library, that you put on there that way. I'm gonna fix it right now.
Can't access.
Let me fix it then. It's not under the right name. Let me fix it real quick.
Oh, I see it from. The link is pointing to the wrong place.
Is it? Oh, sorry.
I'll take care of it.
Okay, you're handling it. I'm not gonna try and edit while you're dealing with it. Two people editing the same document's a bad idea. He's changing the name. There's two that say Illumina. There should be two links that say Illumina now.
I don't understand why it would have worked last week and it did
And
not.
not
It's not
now.
working now.
Yeah, I don't know either. Can we plug your mine in and I could show them on mine?
Yeah.
Try and do that. Because what I'd like to do is show them the users list to make sure that they check they're on here. This is the wrong screen. I am on Dowell on this
Give me
one.
2 more minutes. Just see if it works.
This is what I want to show them is this screen right here. Yeah, 1 second, guys. We're going to show you.
Yep, the document's up there. It is listed under configuring SSH keys.
So if you were not able to follow Lynn's instructions, because we were going kind of fast, there is now a link on the website that says configuring SSH keys, and you can follow the instructions there.
Right here, down at the bottom of the ink class slides.
And again, that will only show up if you hold shift when you reload that page, most likely.
It's not true for me. No?
No. Maybe, other screen. It changed me, so I should be able to do displays now. Displays. Arrangement? Mirror? No, it's not mirror. See if I can just drag this now. No. Let's see him. Why are we not? Maybe. Sometimes Macs have to be closed and then reopened for them to recognize it. Very Windows system. Show this. I can't get this thing to show my computer up there. Do you know how? It clearly changed when I plugged it in because the dimensions changed.
Are you on?
It's on me now. It's hopefully on me now. I guess I could try a different converter because I've only been using theirs. Maybe I try mine and see if it'll let me use mine.
Graham can't log into GitHub because it won't two-factor authenticate because it wants him to confirm he's himself on that until, and it's asking
Phone number?
no, well, it says it was sent a text to, but I think the identifier gave him his iPad.
Oh.
So unless I run home for his iPad, he's not doing anything today.
Well, he can log in as you. You're already on the machine. And all we're doing is Vim Tutor today. So you haven't used Vim Tutor. Let's see. We're going to do this. We're going to do the HDMI to here. Where's the HDMI cord?
You want to go to this, right?
Do I? 'Cause that's VGA. She was using the HDMI. We'll do it this way. Let's see if that works. HD.
That.
Oh, that's why.
Was your key wrong?
I didn't do mine.
Oh, I did that too the other day.
There you go. Now your whole world.
Okay, guys. Thank you.
Now that I got it working.
Okay, guys. Make
You're
sure
gonna have to
real
make
quick.
that like 5,000 times bigger.
I might have to close this anyway.
Do you want
That
me to do
is
mine?
dowel.
Now that you got it working.
That's okay.
I'm on now.
Well, if you want to flip back, it's as simple as moving that.
So just do users. Under users, I want them to check their username is there. Not with the just LS users.
This was just
Yep,
LL
that's it.
user.
Perfect. Okay, guys. Please make sure your name is on this list. I know it's in blue. But make sure your name is on that list. Just a sec, she's gonna change the color. Actually, I'm in white. Want me to do it? Let's do it on mine while you change your color. Oh, you got it? You're fine. Let's try mine. We'll do it on mine. Mine's brighter. I say that, and then I can't get the HDMI into it. Doesn't look that hard. Okay, Command Shift. Okay, please make sure that your name is on this, your GitHub username is on this list. And if it is not, we need to collect your username.
It's not on.
Oh, why is it not showing up? It was a second ago. Computer problems, they're normal for people who work on computers. There we go. Your name should be on that list.
Nope. That's gonna go.
Why is it gone again? Fine. If your name is not on this list, can you give us a red tag? So you're gonna have to use it capitalized then. Oh, wait, maybe it won't work. Well, try it capitalized. If it doesn't work, let us know. Yeah, try it capitalized logging in, and if it does not work, let us know. Okay. Oh, did you get in? Okay. If not, we need to email Matt with the uncapitalized version. Our IT guys. Okay. If you are still trying to log in, let us know. We'll walk around myself. Okay. Let's see if we can get this working. Guys, do not log in under Robin Dowell. You are not Robin Dowell. She is Robin Dowell. Okay, guys, if your username was not up on that board, can you come find me in this corner and give it to me? GitHub username. That's why we did this the first day. You don't need it tomorrow. On a positive note, guys, don't worry too much. We don't actually need the computer until tomorrow. So this is our practice so that we have time to fix it if you're not logging in. Your identity does. I might be because I'm in a lab that does. So you log into that number? I use my identity and that number. I'm like, congratulations. That's quite okay. I don't know why that should. Do you need to make sure it's on the list? Do you want me to see the list?
Okay, so I counted it and I copied it.
We're gonna give the computer time to help, or to put the usernames on there, just give it, and then we'll try again.
Okay, so for those of you that still need help, we're going to help during this section. This is mostly a review of some of the stuff that is in the videos that were for this day, with a little bit slightly different focus in some parts. So I'm going to talk a little bit about library prep and QC. This is also to give you a chance to ask questions if you had questions about the videos. So talking about library prep and QC, we're going to talk about libraries a lot because it's really important in terms of what data you're going to get out. So if you don't have good data, or if you don't have a good library going in, you're going to get really crappy data coming out. So in general, anatomy of a library, there's adapters around an insert. And the P5, P7Ns are specific to Illumina. They attach to the flow cell. You have indices that you can multiplex with, which I'll go over a little bit more later. And then the primer sections, which are just for the machine to amplify or to sequence from those primers. And this is really important, and I know I went over it in the video, but the read is not your entire insert in almost all cases. In most libraries, your insert is going to be somewhere in the few hundred base pairs maybe, and your read is going to be whatever you say it is to your sequencing core. So say it's a 50 base read, or 75 base read, or 125 base read, whatever it is, you're going to get exactly that length out, but it's probably not going to be your whole insert. Again, there's certain libraries that that's not true for, like Micro Rnas, like anything that has really short insert sizes. You might read all the way through, but for the most part, your read is going to be one side of your insert. Or if you're doing paired end, both sides of your insert. In general, creating libraries, as I said, this is not, these steps are not always in this order. Sometimes you fragment. For example, in RNA-seq libraries, there's many kits where you fragment before you do your reverse transcription. So there's certain flexibility in this. But in general, you're going to do your DNA RNA extraction. You're going to do whatever you're doing. So for CHIP, you're going to do antibody enrichment. For nascent, you're going to do run-on reaction, whatever you're doing to your samples in order to get cDNA or DNA out at the end. And then you're just going to have a bunch of DNA. Then you fragment that DNA, or as I said, sometimes you fragment before that, and you can add spike-ins in order to normalize and try and get a little bit better sense of how much DNA you have when you're sequencing, because then it'll sequence the spike-ins as well as your library, and if you know exactly how much spike-ins you put in, then you have a sense of how to normalize certain molecules at the end. Then you add your adapters, again, depending on the kit. You can do that in a variety of ways, and you normally amplify your libraries. Sometimes you don't amplify your libraries. It just kind of depends on your library prep. So I'm going to talk a little bit about kits. Your protocol will determine whether you use a kit. Most whole genome or RNA-seq have a variety of kits that you can find. There's from all different kinds of companies. Again, Illumina's been around for over a decade now, and there are many, many library kits that are specific to Illumina technology. So you can kind of look around in publications for what kits other people used, or ask your sequencing core facility, whoever you're going to figure out what they recommend or what they use if they prep libraries. ChIP-seq, ATAC-seq, more specialized protocols like bisulfite-seq. Many of those protocols do the library prep steps outside of kits for a variety of reasons. So if you're doing something like that, then you're probably going to have at least, you're going to buy adapters that you will add on to your inserts at some point in the process, but you may not buy an entire prep kit. In general, kit considerations are how much input do you have. So if you have a lot of input, whether you have very little input, whether you're doing single cell sequencing, all of those go into what library kit you want to use, what quality input you might have, whether it's really messy or whether it's going to be really clean. Some kits are better for either of those scenarios. Do you have a specific protocol that you're really worried about, either fragmentation or amplification biases? Like if you're really only prepping one type of sequence, then you might need to worry a little bit about what you're using, whether if you're doing a whole genome or a whole RNA type prep. If you're doing RNA-seq, do you want total RNA? Do you want mRNA or poly-A-enriched RNA? Do you want micro RNA? Or do you want mostly total RNA, which is ribosomal depleted? You basically never want total RNA because ribosomal reads, unless you're really sequencing the ribosome for whatever reason, are going to take up most of your RNA. So for the most part, you need to enrich your RNA in some way, whether it's poly-A enrichment, whether it's size, or whether it's ribosomal depletion. And also for RNA, do you want a strand-specific library? Pretty much there's no reason not to do this, and I think almost all kits now do. But that allows you to assign a given RNA to a specific strand of the genome. Okay. In terms of multiplexing, basically multiplexing sticks on certain indices onto specific samples. You pool them all together, and then you can deconvolute which sequences are from which sample at the end. In terms of choosing indices for multiplexing, these are, depending on your application, these are being a little bit more phased out. Single indexing or combinatorial dual indexing, basically you get an adapter or a set of adapters that have an index, and single indexing means that only one of those adapters has an index. So you have only one index on one side of your insert. You're not going to get a lot of samples that way, partially because of, I know that I talked about base diversity. You need to have indices that have certain base diversity. So therefore, indices are 6 base pairs, normally at least, in terms of Illumina sequencing. So you don't have 6 to the 4th possibilities for indices because you can't have any base at any of those 6 positions. You have to have some kind of good mixture of bases at those 6 positions. So therefore, there's actually a much smaller space that you can use. So single indexing is for a very small number of samples. Combinatorial dual indexing is when you have about, 10 different adapters that you on either side that you can mix and match in different ways. I think many libraries are going more toward unique dual indexing, which gives you a lot more possibilities. And then there's some applications for which you want unique molecular identifiers, which basically every single index that you stick onto an insert is unique. And if you really want copy numbers, like if that is the thing that you're looking for, if you're looking for copy numbers in a cell, then you can use unique barcodes for every molecule to determine what is PCR amplification and what is what level you actually got of that molecule in your library. Considerations on this, as I said, base diversity is an issue. You need to make sure that you're not using similar indices for different samples because then the machine is not going to read your data correctly. Index hopping is something that you can read about, but it's where adapters that are free-floating might accidentally attach during the sequencing reaction and stick a different index onto your molecule. And then ease of deconvolution, like unique molecular identifiers, they're great, but it's a pain later on in order to actually try and deconvolute all of those molecules. So those are kind of the considerations of choosing indices. This slide is from the video. In general, these are kind of large categories of issues that you might run into in your library quality control. Contamination, whether organismal, nucleic acid like ribosomal, adapter contamination, base diversity, insert size, complexity, quantity. Those are all, they could be issues, they could just be considerations as you're looking at your libraries. And before sequencing, you can use bioanalyzers, qubits, qPCRs in order to look at all of this stuff, and after sequencing, a variety of tools that we'll talk about this week. Just, also, if you have any questions, please just raise your hand and stop me as I go through this. So pre-sequencing QC, you can use size electrophoresis. What we've used in this building for years is a bioanalyzer, and this is kind of this is an example of a bioanalyzer trace that has several issues with this library. It has excess primers, adapter dimers, and some overamplification. The library itself looks great. This is probably relatively easy to enrich, to clean up, and therefore sequence later on. But getting rid of some of these extra molecules in there is kind of important. Otherwise, you're going to over-sequence those and not enough of what you actually want to sequence. Fluorimeters and qPCRs are good ways to quantify your library. Nanodrops can be off by as much as an order of magnitude. So whatever sequencing facility you're working with probably wants a couple different ways of quantifying your library. They'll also very likely do much of that quantification and the QC in general at their own facility to make sure that they're not wasting your money too. But but in general, don't take that for granted, and make sure that your libraries are good on your side as well.
We've seen people waste 10 grand because they didn't look at this. Because they didn't look at this before they sequenced it. So make sure you look at that before you sequence. And if you don't know how to read it, find someone who does.
So you won't actually see the same concentration among these three different methods, but in general, you can get fairly consistent ratios between qPCR and fluorimeter readings that'll give you a sense for whether your library will sequence well. And as we always say, it's better to make a new library than to sequence a terrible library. You'll waste your money, you'll waste your time. If it doesn't look good, if you don't trust it, then make a new one. All right, so now I'm going to talk a little bit about Fastqc. As I said, we're going to go much more into this later in the week. But in terms of looking at these different these different issues that could be part of your library, there's a couple specific Fastqc plots that I want to go over. One of them is base diversity, and this plot is, the number down here is the position in the read, so this is a 50 base read. And in general, this is a pretty good looking library where mostly it's got similar abundances of each of the nucleotides at every given position. You're always going to see some mess at either the beginning or the end, depending on which read it is. And that has to do with some phasing problems, that has to do with some error rate problems with Illumina sequencing, as well as potential read-through into other adapters. But in general, this is what these should look like for good libraries. This library is fun because if you notice, I don't know if you can see, but green is A. And as you can see, there's a whole lot more A's in this library than anything else. Basically, most of this library is just poly-A stretches. So these people, this is all published data, by the way. So these people basically just spent a lot of money sequencing a bunch of poly-A sections of RNA. And then these are my favorite because this is basically, it looks like a Sanger sequencing trace. You can read what nucleotide is at every given position. This is just an example of a library where people just sequenced adapters. This is the adapter sequence, and they published it. I don't know exactly what they said about it, but they did publish it. All right, so that is one plot that you can look to in Fastqc to look to see if there's any contamination or to see if you have a problem with complexity. Another plot that you can look at is sequence duplication levels. This is kind of a generally good plot from this standpoint where most of the sequences, so this is the sequence duplication level, so one read is where you see most of these sequences, almost 80% of this library, you only see that read once. Sometimes there's a little hump over here where you see some reads somewhere in between 10 and 100 times. You're probably always going to get something. It's never going to be 100% unique. And then at the top of this plot, you can see percent if deduplicated is 82%. So that means that you have about 20% duplication in your library. That's relatively good, depending on your application. This library, in contrast, is probably not going to be super usable. It's less than 20% unique. Most reads, as you can see, are seen more than 10,000 times. I don't remember exactly about this library. I don't know if those are adapters or what, but something happened in terms of over-amplifying this library or a problem with enrichment in this library.
Can you guys guess? Guess what happened? What do you think? You see the same read over and over again.
One thing that just changed a lot.
Oh, yeah. I'm sure that's it. Come on, guys, we're molecular biologists, at least some of us. How do you see the same thing over and over and over again?
And just as a reminder, say this is a 50 base.
If they did not properly remove the ribosome, that would happen.
Yep.
If you over PCR amplified, so you only had a couple of reads and you copied them 10 million times, that would happen. Either way, a bunch of ribosomes of the same read 10 million times, that's a crappy data set.
And just to clarify, this is, say if this was a 50 base pair library, that means you're seeing the exact same 50 bases for most of your library. All right, and then another useful, very clear graph that you can get out of Fastqc is whether you have too much adapter contamination. So this particular plot, again, this is position in the read, so this is they don't really do 64 base reads. I'm not sure why it's showing up as 64 bases. But in general, you don't see any particular adapter at any particular abundance. Whereas, yeah, this is good. This is good. You don't want to see a ton of adapters in your library. Whereas this is probably usable, maybe. It's not as bad as some that I've seen. It's probably better than this. Basically, that base abundance would show up as being 90% all the way across. So this may be usable after you trim the reads, but in general, you see a ton of adapter. You're sequencing adapter way more than you're sequencing most of the stuff that you actually want to sequence. All right. I have scheduled a break in here if anyone needs to get up for 5 minutes
While we're
or...
doing our break, for those of you who still have not been able to log on, if you got logged on, give us a green sticky so we know how many people actually got logged on. If you did not get logged on, give us a red sticky, and we're going to try again during the break. Go ahead and take a 10-minute break, and then we're going to use BibTutor. And don't worry, even if you're not logged on, you can still do BibTutor. So just be a bachelor when I do BibTutor.
Is it going to break down?
Okay.
Did you get it?
Can you give us a green sticky if you are on, so we know you're on? Green sticky if you're on, red sticky if you're not. Anybody still need help logging in that's not available with you?
It's all our standards, you know, the type of.
So if you gave me your username earlier, Matt does have a bunch of new usernames on the system, so go ahead and try it out. Like this. Windows are very small. Make sure you tell everybody to log off so they don't come in through our AWS.
Yep.
Because that costs money.
Okay.
You might as well do it on your own laptop or
All right. No, she's gonna try it later. All right. So, most people are good. If you are on the system, then please.
Okay, guys, we're gonna start up again. If you still have trouble, give us a red tag so that we can help you get on. Otherwise, follow directions up there. We're doing good tutor.
All right. Hang on. If you are on the AWS machine right now, type log out. You don't want to be on the AWS machine in order to do Vim Tutor. You don't need to.
By the way, guys, what we did today, setting up your SSH keys, you should never have to do again.
Yes.
Just do that once. From now on, it's just username and number. You're done.
Always a drink. That's the right.
Really big. All right. Hopefully this is readable to everybody. So when you're on your terminal, just type Vim tutor. I'll get out of there. Just type Vim tutor and you will get into a big long text document. Vim tutor, no space. Guys, we're starting, guys. All right. Vim tutor, no space. Vim tutor, no space. And that gets you into the Vim tutor. Mine is really big so that hopefully everybody can see. Most of these lines are just things that you can read later. So this just is an introduction to Vim. Actually, I have a slide. That's a brief introduction to Vim. Whoops. So Vim is a text editor. It allows you to read, write, and save text files. It's entirely keyboard based. One thing that people get hung up on is you cannot use your mouse to move the cursor. You have to use your keyboard. You can click places, you can highlight words to copy them with your mouse, but otherwise you have to use your keyboard and actually to move your cursor where you want to type. All right, that was the only intro I had. So most of this you can go through by yourself. In general, I'm going to go through the first few lessons with you. First lesson is moving the cursor. I have to be honest, I use my arrow keys. You can use your arrow keys or you can use HJKL. Whichever, if you want to be fast, then use HJKL. If you want to be slightly slower, you can use your arrow keys. So that's kind of a personal preference type thing. I'll let you read most of this by yourself later because you're going to do some of these things for homework. Exiting Vim is probably the most important thing you need to know. I guess to step back a little bit, there's two modes in Vim. One of them is what they call normal mode or command mode, and that allows you to change general properties of your text file, but not to insert text. The other mode is an insert mode, where you actually type within your text file. So, unless you have a little thing down here that says insert, which I do right now, you are not in insert mode, you are in command mode. In command mode, you can type semicolon Q. If you have not changed the file, this is enough to quit. If you have changed your file at all, you need an exclamation point to say that you don't want to save any of the changes that you've made. But that allows you to get back out to your command line. Just :Q. So if you ever are confused about what you've done with your file, :Q, exclamation point, means that you will reset to when you originally opened that file. In terms of editing, so it wants you to go down to this arrow line, and you can press the X key to delete. You are still in command mode, you're not in insert mode, you can't type anything in this mode, which is why X doesn't make an X, it deletes. So you can fix all the typos there. It's going to feel unintuitive at first, but you'll get used to it.
At this point, we are just following the Vim tutor exactly as it's written. So if you want to go faster, just read the Vim tutor and do what it tells you to do.
All right, now we're actually going to type some text. So you can press I to get into insert mode. You have a little insert at the bottom left of your screen that tells you that you are able to add text. And then you can just add text like in any word processing document. In order to get out of insert mode, actually, I guess I didn't do this the way Vim Tutor wanted me to. In order to get out of insert mode, you press escape. And then your little insert will be gone from the bottom left of your screen. You'll be back in command mode. You can delete with X. You press I to get back in insert mode.
Just for a second, practice going in and out of insert mode and changing stuff. That's the part that's less intuitive for people who've always used Microsoft Word. In Word, you're always in insert mode.
All right.
For those of you who like history, this Vim was one of the very first word processing systems. It's like 50 years old. It's probably older than most of the people in this room.
All right, so the I is one way to enter insert mode. The other way to enter insert mode is A. The only difference is where it sticks your cursor. So I have my cursor right here under this period right here. If I press I, my cursor stays exactly where it is. If I press A, my cursor goes one space forward. So this is append instead of insert. They're functionally not very different. But sometimes, especially if you're at the end of a line, it just makes it a little bit faster to append so that you can go further beyond that text than to insert. All right, and outside of just getting out of Vim, the second most important thing, well, maybe insert is the second most important thing. The third most, because if you can't insert, then you won't have anything to write. The third most important thing is saving your file. You need to be out of insert mode, so I'm in insert mode right now. If I'm in insert mode, if I type a command, it will actually type it. Oh, I guess I'm a little bit off the edge of the screen. There we go. If I do,
insert mode right now.
if I do :W right now, then it's just going to type it in my text file. That's not what I want to do. I want to escape out of insert mode. And then :W is going to write to this file right here, down here. So it says that it's written. So everything that I have changed about this text file is now saved in this file right here. If you want to just write and quit, just save and quit, then you do :WQ, because that does the :W, which is the writing, and it does the :Q, which is the quitting. And that just gets you back to your command line.
you should make them do their summary by themselves.
Yes, no, I'm gonna have to do the summary. So, your homework is to play around with this until you feel comfortable, do the summary, and then I don't remember what you assigned. I
You
think
pretty much
section
need to
2.
know all of the commands in lesson 1 and 2. Beyond that, the lessons 3, 4, 5, 6, I think it goes to 6, maybe 7, 8, either ends in 6 or 8. Those are all optional to know. You do not need to know them. The nice thing about doing the entire BIM tutor is not that you will memorize everything in lessons 3 through 8. That's not going to happen. I don't even know everything in lessons 3 through 8. Zach and Robin like, but everybody else won't. What's nice is you know it exists. So if you get to the point where you get really annoyed because you can't get to the end of the line very quickly, you'll go, wait, Vim had a command for that. And you will Google Vim end of line. So don't worry about memorizing beyond lesson 1. Do memorize all commands in lesson 1 because you will need those this week badly. But beyond lesson 1, you probably don't have to memorize them. You just have to know they exist. So that when you get annoyed, you can go Google them and become a faster coder programmer.
I'll be totally honest, I use about 3 commands outside of lesson 1
Yeah.
on a regular basis.
So lesson 1 is the one to memorize all commands in lesson 1. Beyond that, you don't have to memorize them, just know they exist.
Okay. Anybody have any questions about Vim right now?
You do not need to log on to the server to use Vim, and actually we would prefer you don't, because we're paying for AWS off of a grant. So if you go onto the AWS, it actually costs money, whereas if you're using Vim Tutor on your computer, it does not. So just use Vim Tutor on your own personal computer, and then when we use the AWS is when we need to use the AWS. Okay. So
Okay. So, I'm going to do the second part of my sequencing overview. I just wanted to break it up a little bit. So talking about sequencing technologies, there are short read technologies and there are long read technologies. The short read ones, these all work on slightly different chemistry. They give different outputs. Illumina is by far the most popular, and therefore most stuff is built for Illumina outputs. But there might be some of these other short read ones that might be more useful for your application if you're doing something a little bit different. Long read sequencing, there's a couple right now, and I think they seem to be getting better all the time. There are, especially if you're doing anything involving genome assembly, which is not something we're going to talk about in this class at all, long read sequencing is quite useful because assembling a genome in 250 base chunks is very difficult. Assembling a genome in 50 kilobase chunks is going to be a lot easier. So if genome assembly is something that you're doing, then you might want to look into these long read, but we're not going to talk about them in this class.
And there's a class over at eBio for it if you want to take a course.
All right. So I'm just going to go on to... There are many YouTube videos on Illumina sequencing. I'm just going to give...
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow.
So this is specifically for paired end sequencing. So that section that they just talked about, if you're only doing single end sequencing, that's all that happens on the flow cell. And then if you're doing paired end sequencing,
Cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire
does anyone have any questions about that process? Pretty clear.
So, important points to remember. All they're really doing is taking pictures of glowy dots. Okay? Glowy dots must not be too close together, because if they're too close together, you can't tell them apart. That's what's called overclustering. That's why it's hard for sequencing centers, because they're trying to get you as many reads as possible without overclustering. So it's this load just enough but not too much problem. You know, Goldilocks. Okay? What you get off the machine is the glowy dots. That's it. They're giving you the glowy dots.
Whether or not the glowy dots are right.
What color is green? What if I have a very light green? Is it really green? What if I have a very dark green? Is it really green? The computer has to do that every time. It has to figure it out. So later this week, we're going to talk about the read and then the read quality. And that's how good do I think I guess at the colored dot. All you're doing is analyzing millions of colored dots.
When the channels are flooded with the fluorescently tagged nucleotides, is it like a bunch of G's and a bunch of A's, or is it all of the nucleotides?
It's just a mixture. Yeah. And actually depending on the platform itself, depends on exactly which nucleotides are labeled and which color. So the different machines.
Your major difference between those machines is really what dots are we going to be coloring what colors.
Yep. Anything else? All right. So these are,
Anybody ever wonder why it's cheaper to use one of the bulk systems? I call that like going to Costco for toilet paper.
yeah, so this is, in general, if you don't want a lot of reads, you can use, something down on this end, but technically you're paying a lot more per read because it still takes a certain amount of money to, fund the machines and the chemistry and all of that. Whereas these systems have larger flow cells and are just able to image a whole lot more at one time, and therefore it's going to be less money per read that you have.
The NovaSeq actually only uses 3 colors. So, well, how do they use 3 colors instead of 4 if we've got 4 nucleotides? No glowy dots, that's right. No glowy dot is the 4th color. Well, what do you think that means for your probability of being right? Right, your probability of being correct at any one base goes down. So in the NovaSeq, you've got lower probability you're right, but way cheaper because you've just thrown away 1/4 of the fluorescent dots. And those fluorescent dots cost a lot of money.
Yeah.
Also means that when it fails,
I didn't see a glowy dot, it must be a G.
Yeah. So this is the difference between the 4-channel, 2-channel, and 1-channel chemistry. In terms of what is tagged what. So for the 2-channel, A is tagged with both, T and C are tagged with 1, and G is not tagged. And then they can deconvolute that at the end. Yeah. No, this is kind of an average for what they're able to output. So if you have a little bit higher cluster density that they can actually deconvolute, then you might get you might get more reads out of it. So it kind of, there's a maximum where it's like, if you have more clusters than that, there's no way you're getting data. So it does have a maximum that you're ever going to get, but if you get a spec of like, you're probably going to get 350, 400 million reads off of this machine and you get 425, that's not a problem, necessarily a problem with the extra 25 million. Does that make sense? Yeah. Okay.
That's a good question. So if you repeat the question, how do you know if you have over clustering? So it tells you there about how many reads you get. The more optimal you were, the more higher you'll be on that scale. So for instance, the next seek goes up to 400 million, right? So if you were perfect about how much you load, that's how much you get. If you loaded less, you'll get less reads. If you load too much, you get less reads. So why do you get less reads if you load too much? It's not clogging the machine. Remember, we're taking pictures of colored dots. So what happens if you're taking pictures of colored dots and you put too many in there? They can't discriminate between them. They're too close together, and I can't tell this read from this read. And so every time I go to guess, I get a green and a red. And it's like, crap, I don't know what that is. It's a green and a red in that colored dot. It's because they were too close together, right? So the machine, the first thing it does when it gets those colored dots off is throw away anything it couldn't tell apart.
So the onboard software does a lot of QC before any of those reads get to you.
Yeah, Jacob, what were you going to add?
How many molecules represent a single read?
Oh, it's thousands. I don't know exactly how many, but it's thousands. One read, why do you have to have thousands there to see a single read? Why do they have to even do PCR on the cluster? That's right, to make the glowy dot bright enough. So one copy is not bright enough that a camera could take a picture of. So you have to do PCR, so you have thousands of the same copy right in the same position, so that it can make enough green glowies that you can tell that's really green there. They do, because they throw all the colors in, take a picture, and then wash that green dot off. So they actually cut all of the colors off after they take a picture.
Because they need to excite them all at the same
Yep.
time.
So they take a picture that excites them all, and then they cut them off and add the next nucleotide. This camera in there, honestly, is not that much better than an iPhone camera.
Yes, it's not really a microscope.
Yeah, it's kind of crazy that type of camera is what's They have, but it's not cost effective. So it's cheaper to do more chemistry, more PCR than it is to improve the camera. No, one picture is one cycle, one nucleotide. So you take a picture, you wash off all the colored dots, do the next nucleotide, take a picture.
Well, you take several pictures usually.
Now, the
So,
old machines actually saved every picture and you could get a copy of it, but the size of that data was like terabytes every time they take pictures. So they have, now the machine, unless you force it for troubleshooting, will not save those pictures because it's just very expensive to save that many pictures. Yeah, so
Right,
one
so this is
thing
massively-
all cycles. And one of the things you will see in your QC stuff on Thursday is there's actually a picture that tells you the nucleotides at every cycle. Because let's say you flood the machine with all 4 colors, but something goes wrong and only the red one gets in. You're gonna get nothing but red dots for a cycle. And the computer goes, what? That's not possible. And it's for the haywire fix. And you will get a weird symbol in your, I've only had that happen once in the hundreds of times I've sequenced, so you're probably not likely to see it, but it shows you colors per cycle so that you know if any one cycle went haywire.
so this is basically Sanger sequencing, except millions at a time. So it's massively parallelized Sanger sequencing in a lot of ways.
And the height, if you've ever done Sanger sequencing, the height tells you how good that glow was. That's what our quality is going to be, basically.
Yeah.
How good was our glow and how sure, have you guys ever done a Sanger sequencing where you have something where there's 2 nucleotides in one position and you get like 2 half humps that are green and blue at the same time? Like if you have a mutation, that's what happens. That's that quality score. That's going, okay, I think it's green, but I kind of saw some blue there.
So the onboard software does actually output a lot of QC metrics. So if you need to troubleshoot with your sequencing facility, like something that might have gone wrong with sequencing versus what might have gone wrong with your library, then a lot of those QC metrics might be useful. Most of the time, you probably won't look at it. Once you get your data back, you'll instead do QC on your library, and if everything looks like junk there, then you can try and go back and troubleshoot. Was it the sequencing? Was it the library? That kind of thing. Yeah.
Let's just say we're really big on quality because we've seen too many people analyze data for 4 months before we ask them if they ever looked at whether their library was crap, and they didn't. Check that first, please.
All right. So I also did a video about designing a sequencing experiment and kind of looking at this pentagram, I guess, of considerations for sequencing experiments and talking about read depth in relation to all of these other things. And in addition to read depth, the things that are kind of particular to a sequencing experiment versus any other type of experiment are insert size, read length, and single paired end. So I just wanted to go over this a little bit in case anyone had any questions about it. Again, this is mostly covered from the video, but I added on some of that other stuff. So if you're doing RNA-seq, just kind of basic differential expression analysis, you can get away with about 20 million reads per sample. You can do it either single end or paired end. Basically the benefit of doing something paired end is that you might get a little bit better mapping, or you might trust your mapping a little bit more doing it paired end, but it doesn't matter all that much in terms of just basic differential expression. And then your insert size should be in the hundreds, somewhere between 200, 800, something like that. If you're doing low abundance RNA, you want a lot more reads per sample because you're not going to be picking up, statistically speaking, you're not going to be sampling enough of your reads at 20 million reads in order to get some of those low abundance RNAs that only have, say a couple dozen copies versus hundreds of copies in the cell. You can also do those single paired end, insert sizes should be the same. If you're doing isoform analysis, if you really want to look at splice junctions of RNAs, then you need, again, these are as I said in the video, these are bare minimums. You really probably want more than 50 million reads per sample for this. You probably want more like 100 million reads per sample. And you do want to do them paired in so that you can see both sides of the splice junction. Longer reads might also help because then you might actually overlap more splice junctions, just statistically speaking. For whole genome sequencing, for general homozygous mutations, you might want 15 times coverage of your genome per sample. You can do that either single paired end. Again, the benefit of paired end is that you have a little bit more trust in your mapping, but it's going to be generally more expensive. But you can do it either way, basically. If you're doing heterozygous SNPs, then you want a little bit more coverage just to have more confidence at any given position that you're seeing both nucleotides there. And doing indels, just in terms of trying to build what those might be, you want a lot more coverage than that. And you want to probably do those paired in so that you get a little bit more sense of, indels or different kinds of mutations like transversions and things like that, what both sides of those might be. For ChIP-seq, you can get away with fairly low numbers of reads per sample if you're just kind of doing basic ChIP-seq. Generally, there's no reason to do paired end in ChIP-seq. You can do single end. If you want broader peaks, if you think for things like histone modifications, things like that, then you might want a little bit more depth in order to get better sampling across more of the genome, basically, because if you're looking for broader peaks, then you're probably looking for more of the genome. Yeah.
What is the distinction that makes that for ChIP you wouldn't want to do paired end?
Especially if you're doing narrow peaks, there's not that much of a reason to, because you should be getting enough sampling of anything that's enriched from a single end of your insert.
Paired end costs more.
Paired end costs,
And you don't gain anything.
yeah, basically, yeah.
You're already enriching with like IP pull downs, right?
Right, right. So the trade-off on single end versus paired end, if you want more molecules, then you want single end. Like if you want to actually sequence more molecules, if you want to sequence half the number of molecules, but from both sides, then you get paired end. Does that make sense?
You only do paired end when it gains you something, and it gains you something in RNA isoform analysis. It gains you something in whole genome if you're trying to glue it back together, because you've got two pieces that are far apart, it's easier to glue everything back together. And it gains you something in ATAC. If it doesn't gain you something, you don't spend the money.
Jacob?
Single end versus paired end doesn't impact your choice of insert size, or like how you define your insert size distribution?
Well, for some, not for any of these that I've shown so far. If you think that your insert, so say you're doing 50 base reads, if you think that your inserts are going to be 50 bases, then there's no point in doing paired end sequencing because you're just going to sequence from one end 50 bases and sequence from the other end 50 bases and get the same exact readout twice, which isn't going to help you at all. So for any of these particular libraries that are going to have insert sizes in the hundreds, you probably wouldn't want to do 250 base reads paired end. You're going to get a lot of the same sequence because your inserts, say roughly half of your inserts are going to be less than 500 bases. So then you're going to read into the same section, and that's just kind of a waste of money. Does that make sense?
By the way, a sequencer will not, at this point, and I say at this point, it's been changing, will not go above about 1,000 base pairs. You cannot go above that at this point. However, when I started sequencing, don't ask how long ago, the biggest insert size was like 100 and the read size was 20. So it's going up.
I do want to, something that I,
Now, yeah, 1,000 base pairs is the longest that you can PCR.
For like an
And the read length, I think the longest right now is 250.
yeah, 250 is the longest. And your read length, you could go, it depends on the machine, first of all. So whatever facility you're going with, check what their machines are, check what they recommend. Generally, if you're doing really short reads, you might, depending on the quality of your data, you might start running into mapping issues. So 37 is the lowest that they do right now. Most things above 25 base pairs should be uniquely mapped to the genome unless they're in repetitive regions. Depending on your read quality, depending on the quality of the data that you get out. So the length of your reads is, I think, it matters, but in a lot of ways it doesn't matter as much as these other things. It's more machine dependent. And then in the video I did high C, but I figured I'd do a contrast for Micro Rna. Generally Micro Rna, since you have a lot smaller pool, so Micro Rna you would do a size enrichment in your library prep. You're going to have a smaller pool of RNA, so therefore you don't need as many reads per sample because you have a lot smaller pool that you're sampling from. You're never going to want to do that paired end. You're going to want to do short reads, and those are going to be very small insert sizes. So that's just kind of a contrast to all of these others.
Do notice that this says in mammals.
Yes.
In E. coli, you can get away with hardly nothing.
Yeah. So E. coli, you would, going back to this, E. coli, you would never, ever, ever sequence on a next-seq unless for some reason you were multiplexing. Yeah, basically. So E. coli, you're going to use MySeqs, or iSeqs, I guess, to sequence. So these larger throughput machines are for larger genome creatures. Does it? I don't know.
I didn't know that either.
I think it does.
Okay, well this is, I cited my source, which is
So
old,
Taylor says that Geo
but-
apparently has standards now up there.
yeah. I would follow theirs. Again, this one's old.
Actually,
I just
go
kinda grabbed it.
further, Yeah. I tend to go higher than the recommended limits, just.
Well, this is not recommended, this is minimum, bare minimum.
Recommended.
Yeah, because I think
Because remember, when you load on the machine, do you have any?
I'm
guarantee
sure.
you're actually gonna get 10 million?
Yeah. No, this is not best practices, this is bare minimum. Like, you might be able to get something out of these.
If Amber is running it, but Amber moved to Illumina, that was the person running the core here, we liked her. But you have no guarantee you will get this money, because get this number of reads. When you say you want 40 million, you're gonna get around 40 million.
Also, if you're multiplexing and you have, say, 12 samples in a next-seq run, some samples you might get 35 million, some samples you might get 12 million. It just kind of depends, like the pooling is not perfect either. They do the best they can, but it's always going to be variable exactly how many reads you'll get out of a multiplex experiment.
And if all that isn't bad enough, not all your reads will match.
Yeah.
Just, if your PI is pressuring you to go as bare minimum as possible, ask them if that's how you'd like to run the lab too. I'm serious, they want to go cheaper, but if you go cheaper, you might not get what you want out of it, and then it's useless. It's wasted.
Something I will mention is sequencing for greater depth. If you have a library or a set of libraries that you sequence and you don't get the depth that you need for some samples, then you can go back and sequence again, and you should get another set, sample from that library, and hopefully you'll be able to just add those two together.
So generally, when you make a library, you're going to have 100 to 1,000 times more material than you need. Which means if you do sequence too low, you just give it back to the sequencing core and they run more sequencing.
All right. That's all I
The
have.
other tip I have, now that we don't have Amber downstairs, I never give my entire library to anybody. When I make a library, I split it in half and keep half of it. Because if anything happens on the other end of the core, I want to have my backup.
And I know this sounds like something I shouldn't have to say, but don't sequence again if you didn't use the
please don't, no. Check your QC before you resequence. And actually, on one of these days, we will cover a program called Pre-Seq, which is awesome, because what it does is it says, if you sequence more, will you gain anything, and if so, how much? And it will literally draw you a line that says, this is how much you sequence. If you double how much you sequence, this is how much information you'll gain. If you triple, this is how much you'll gain. So they draw you a line to tell you, you cannot gain any more than getting 100 million out of this, or whatever it happens to be. It's a great QC program, because it tells you how deep you could go before you stop gaining any more information.
All right, does anyone have any other questions about library prep, like pre-library or pre-sequencing QC, post-sequencing QC, again, we're going to go over later in the week in more depth. Sequencing, cool.
Okay, last thing we're going to do today is logging into X2Go. If you did not download X2Go, this will not work. It was in your pre-class instructions to download X2Go. If you would like to do it on your own, on the website, is where we're going to pull the slides from. So we're not even going to do slides other than the ones that were on the website. Just a second. My computer has suddenly frozen up. Like completely. Do the Microsoft thing, restart it. I got nothing. Lynn, can we use yours? Because I am totally frozen. Yeah?
X2Go is separate from X2Go,
Right, so X2Go, we just said that you're going to do all this typing and you can't log onto a computer with a GUI. We were half lying. The majority of things you do on a supercomputer, you cannot do. Oh, look, I'm finally clicking. That's okay, it's just going to the website. Okay, so you're going to go to X2Go. The majority of things you do on a supercomputer, you cannot visualize. You will only have that little black box, little white box and typing, okay? Except that we have set up a visualization system. We have the most awesome IT ever in BioFrontiers. If you're not in BioFrontiers, I feel bad for you because we have the best IT. And he set up a system so that you can actually log on to a supercomputer with a GUI when we need to look at files. So this is only for looking at visualization files. We will not do that until Thursday. But again, we must test that everybody can actually do it. So we're going to set it up today just to make sure we can set it up. On the class sequencing day, there is a file that says using X2Go to log onto a supercomputer. So go to that file and attempt to follow the directions. If you get stuck, give us a little red sticky. I'm not even going to do it up on here because I think most of you can just follow the directions, but give us a red sticky if you can't follow the directions on using X2Go to log onto a supercomputer. If you have not yet logged onto that supercomputer, it is not going to work. This is not the right number, okay? This is the normal AWS that you will be using for most of class. There's a different number for the biz node. And so the biz node number is actually in the pictures, but I'll put it on the board anyway because it's super, super tiny. This one right here has got, can I zoom in? Control, Shift, Plus. Does that work on this one? Yeah, so if you look there, the number is right there.
That's not up there.
Oh, thank you. 3 dots. That. 15, that's the number they want. Oh. Everybody says my 5's quick guesses. So follow the directions on the website. If you get stuck, give us a red sticky.
.250.
Thank you. So this is only for visualization. So you'll log on to that one if you're in the terminal. This one you only use for the X2Go. I should note, by the way, these supercomputers are only up for the 2 weeks of the workshop. It will die on Monday, so do not put data on this thing.
Mary, what are your
Not big enough? Okay, I'll make big ones. Big. But the instructions are in that document. If you get stuck, raise your hand, or not raise your hand, push your big sticky note up. Once you get onto the machine and actually see the IGB window, then put us a green sticky note so we know you got it done. And if you have a green sticky note right now, can you pull it down so we know you're not done? There you go.
Yeah, we did on here last time. That's our studio. I don't think she did this.
Oh, you know what? Is everybody getting this there? Matt may have shut the machine down. So this is the expensive machine, which means he may have shut it down. So if nobody's getting in, it's probably that he shut that machine down. You got into Xgo? Okay, insufficient memory is fine. That means he's not the problem. No, someone got in, so he can't get in. Okay. But if it says insufficient memory, that's okay. It just means that the machine's not there yet. Wait, no, I have seen this before. Okay, so I've seen this before. This is really stupid. It's Starbucks going. A long time ago, I had that airport and an old computer. The only way I could get it to go was Starbucks. Something about the computer that pointed to the wrong position. Like it's not pointing to, and it's a Windows computer, right? It's not pointing to the right. So start it on the and if that works, we'll probably send it. I had to do that. I remember that now. Yeah. So this Xgo has a pointer inside of it that tells it where all of the SSH stuff is, and it's in the wrong place. So you can fix it, but it's not. Click again. You can just launch it from the like that. If that does not work, let me know, because we're gonna have to reset your pass. What happens is that the system. Yes, so what happens is it's in the wrong place. It's looking in the wrong place for the Is everybody ready to
Hell yeah, actually the Macs almost always just work way better. Oh.
tell everybody that?
Yeah.
So we're gonna pause. For those of you who are having trouble getting on, don't worry, like I said, we'll use this till Thursday. Can you tell me if you're having trouble? I just want to see, are they all PCs? Do we have any Mac users having trouble? Okay, some Macs, okay. So I think I know what it is. There's some hidden setting in Xgo that's not pointing to the right key. So even though you check the box that says point to the key, it's not pointing. So that may take us a little while to fix, but we don't have to fix till Thursday, so we're okay. I want to do one more thing before we go today. We'll come back to that. So now that you know the very basics of Vim, we are going to do something called setting a variable.
Want me to do it?
Yes, just. Okay, setting a variable. When you are in this little square box, you are typing in what is known as Unix or Linux. So if you ever need to know how to do something, you Google Linux setting a variable or whatever you want, right? So what we're going to do is set a equal to 5. Most important thing about Bash is it does not like spaces. Do not put spaces in that. It will tell you it's not true, okay? A equals 5. Yeah, that's bad. A equals 5, no spaces. Whereas A equals 5 with no spaces works. There you go, okay? So what happens now if you type A? A not found. You have got to call a variable to use it. So to call a variable, you put a dollar sign in front of it. So dollar sign A, oops, I should have done it.
It says 5 now, so you just basically
Oh, look, you did, look, 5. Who?
echo.
Echo.
Okay, right,
Echo, dollar sign, 5.
A.
A, thank you. There you go, echo A. Echo is how to tell you something, and you told it to tell me what A is equal to. Okay, so without Lynn doing it, set B equal to 6. And then echo B. Echo dollar sign B. Now, try echo dollar sign B and try echo B. What happens? Okay, so if you give it the variable without the dollar sign in front of it, it just gives you back whatever you told it. If you give it with the dollar sign in front of it, it gives you the variable, okay? Now, C is equal to A plus B. Again, it does not like spaces. Do not give it spaces. And then echo C. Why didn't it work? Ha ha! You did not call the variables. How do you call the variables? Don't show them. Make them do. Oh, they did.
Sorry.
Dang it. And now echo C. Okay? Now, what I want you guys to do, you have learned the very basics of Vim Tutor. Okay? We're going to open a file. Let's make it vim add.tx or add.sh because it's a shell script. Add.sh. Okay, that opens a new file named add. Okay? I would like you to set A and B equal to 2 numbers and C equal to the addition of them, and then have it echo C. But make sure you use those dollar signs, because if you don't, it is not going to work. And she's not going to do it for you, so we're going to see if you can do this. A equal to a number, B equal to a number, C equal to A plus B. Don't forget your dollar signs, and then echo C. Remember, you're in Vim, which means you must be typing in insert mode.
It's not going to evaluate.
What's that going to evaluate?
No.
Until you put little parentheses around that,
Yep.
because
Yep.
so, just so you know. I don't know if you were trying to.
We're doing it one step at a time to get them to logic through it.
Okay, all right. I just wanted to make sure you knew that I was just going to put 5 and 6. So.
We're going to do it one step at a time so that everybody remembers. The problem we always have is nobody remembers which side the dollar sign goes on. Dollar sign means I'm calling this. Okay, unplug your computer and do it. So they can't see what you're doing.
Well, I could have, whatever. I can bring it.
Oh, you could just hide it. That'll work.
I'm extended here. Just like this, or
Yep,
with it
and then,
evaluated?
no, you're right. And then you need to do just like this for now, because we're going to show them in a minute what it looks like. Yep, right quit, and then bash and or that. Okay, then go ahead and add the brackets. Oh, we want to delete it though. We'll do it afterwards. We'll show them first.
No, it's okay.
You could actually put it there. You're smart. If you need help, give us the red sticky. If you don't, make sure your red sticky's not up there.
What is it? I actually don't do math in Bash.
Oh,
ever.
it's just parentheses.
Just parentheses around the whole thing?
Yep.
Like that?
Yeah, I think so. Go ahead and echo C. I think it might actually have to be around the letters themselves. But we'll find out. And then evaluate it. No, it's around each letter individually.
Okay. Like that?
I think the parentheses go on the inside of the dollar sign.
Like that?
Yeah, I think so. I'm
I
pretty sure
have
that's it.
never done this. I've done math with block, but I've never done it just on bash.
Oh, it's not adding. Oh, let me look it up real quick. It's the curly brackets. What am I thinking? It's curly brackets. Sorry. I'm stupid. It's not regular brackets, it's curly brackets.
No. I mean, I did 5 plus 6. The curly brackets around the plus?
Maybe. This.
Oh, double parentheses,
Double parentheses. Or square brackets.
square brackets?
Yeah, it lets you do it square, with the dollar sign in front of it. Yep, square bracket, dollar sign in front of the A. I mean, dollar sign in front of the A and B, though.
Nope.
Oh, you're right.
It's not with square brackets.
Yeah, looks like it. I think you have to have dollar signs there. Nope. Okay, try it this way. Old deprecated also works right there. Two parentheses. There you go. That should work. There you go. Okay, guys, are you ready to see if you got it right? Let's do what I told you to do first, because I wanted to show you it does not work this way. Okay. So you can set A equal to 5, set B equal to 5, and try and add them. Go ahead and run this.
So the way you run it,
Remember, right quit to get out, escape right quit.
come on.
So escape WQ to get out. And then in order to run it, you need to do SH, whatever you named it, okay? Or bash, whatever you named it. I always use bash. And it gave me 5 plus 6 because that's literally what you told it to give you. Okay, so now if you actually wanted to add, you need to go back into your program and add double parentheses around it. So it should be set up like that.
So this is a bash syntax thing.
Yeah, so go ahead and close that again. When you run the script, you type sh space add.sh, or whatever you called it. If you named it add.sh.
In this case, sh is a command, and it's acting on this file, which is something you'll talk about more tomorrow. It is a plus.
It is a plus. So it's how you tell it to evaluate it.
Yeah.
So I don't know that you actually need
I
that.
don't-
I just put that there. That means,
See if it works.
yeah, let's see if it works without it. I don't know that you need that actually, but it makes it glow green, so you know.
So you know.
You do.
Yep, you do.
Pros and cons.
Yeah, that's basically what it is. If you don't know how to do this, all you do is Google add in bash, and somebody will answer the question. So what's the dollar sign mean there? So dollar sign means evaluate this. So A is A unless you evaluate A. And when you evaluate, check it. Or you pull it out of memory or something to that effect. The dollar sign means evaluate this. Congratulations, you have coded.
So, next to go is asking for a password.
Did you see when you set up your settings?
So are we there now?
Just talk about variables.
If you have evaluated and.
Hang on. So we will not actually, whoops, see, I'm in insert mode. We will rarely be actually evaluating arithmetic in Bash. Mostly your variables, just as, you know, we'll talk about this more about this tomorrow, but mostly your variables are going to be paths to certain files or file names. So like, if I wanted to, whoa,
Well, look at that private key.
Indeed.
all right, so,
Oh, this is important, and it doesn't teach you this. Oh, this is important.
What?
Okay. If VimTutor ever gets stuck or Vim ever gets stuck because your computer crashes or you close it in the middle of it, we need to show them how to do that. Go ahead and finish what you were doing.
Okay.
We gotta show you how to do that, 'cause otherwise it confuses the crap out of you.
so basically, there's a lot of times that you're going to be copying paths and sticking them into variables. So, you know, PWD is wherever you currently are. If you copy that path and I say A equals that path, then I can call A and it'll give me that path. That is what we will mostly be using variables for. I just wanted to, we're mostly not going to be talking about, you know, doing addition. We're mostly going to be doing paths within variables so that if you're going to the same folder, the same directory all the time, you only have to reference the variable and not a long path.
You had a question over there.
Yeah.
If you exit out now and you go back in, would it still be saved?
So she said A on the command line?
Oh, if I exit out of my terminal, A will not be saved. There are ways that you can make global variables, but that's a config, that's when you open up Bash, then it can set variables for you, but that's a different story entirely.
Yeah, we're not gonna go over that. It is possible to set A to something permanently. We are not going over that in this class.
All right, so I'm going to make a file. Whoops.
Yes, we're gonna pretend crashing on purpose, because this happens to people every year, nobody knows how to get around.
All right, I'm going to make a file. I'm going to put gibberish in it. I am going to
Purposefully crash it.
close out of this terminal.
She has just purposefully crashed her computer. Now she's gotta go get it back open again.
So what happens, so
Go ahead and try and open it first, because this is what they'll
Okay.
see first.
what you'll see is there's not actually a file there because I didn't save it. There's just a swap file.
Okay.
But
But go try and open it, it'll still give
Yeah.
you the error.
so if I,
If she types, but you can't see that, it's down at the bottom.
if I try to open that file, you're going to get something that looks like this.
That
And
means
you're going to say, what the hell? What is this? Basically, when you're editing a Vim file, it creates what's called a swap file. So that if you want to exit out, it'll just delete the swap file. It's basically kind of like any, like Word, it has files in progress, you know? And if you crash out, it says, oh, do you want to recover this file? This is the same exact thing. It's just a little bit confusing how it's worded. So you can do a variety of things with it. In this case, it's actually a little different because I didn't save it because it can't revert to anything. But you can quit out of it. You can delete your swap file. So I could delete this swap file right here.
Which means the file goes away. You may or
Here.
may not want the file to go away.
Let me get what they'll actually see though. Write that.
saved that one.
I saved that one. This is what you'll actually see. Sorry about that.
If you save.
Yeah, you will almost always encounter this with a file that already exists.
In the middle of typing a file, if you want to save, :W, or escape, :W at any time will save it. And just like Microsoft Word, save all of it.
All right, so now you're gonna see my pretend cr.txt. That was my original save that I saved, and then I have a swap file. So if I try to open this up, you're gonna get this. You can do a few things with it. This is so large. The ones at the bottom are what you can do with this file. You can open a read-only version, which means it'll keep both files. You can edit this file anyway, which one does it open in that case? The swap file?
Swap file.
The swap file. You can recover, which goes back to your original file. You can delete the swap file, or you can just get out.
Would it edit anyway, open the version of the file on disk and recover a copy of the swap file to the...
I don't know.
I think
Yeah,
E
so
does it.
E opens the file on disk no matter how it exists. R is going to copy the swap file to your original file.
Okay, yeah.
Just in summary, .swap files have the like auto save feature.
Yeah, that's
Yep.
kind of what it is.
Yes. Swap files are the autosave feature of DIM, the original autosave.
The original.
All right.
So,
I'm gonna say
pick one of those options.
recover, and that gets the one that I edited after I originally saved it.
Now,
Okay.
it will continue to do that for the eternity of this file, unless you delete that swap file. Okay? So you must delete the swap file in order for it to stop doing that to you.
Which we'll go over tomorrow.
Yep, we will talk about removing files tomorrow. I just wanted you guys to know what it looks like if it crashes, because that confuses people every year.
All right.
So right there is how she deleted the one that ends in .swap.
And never recover a delete.
Do not delete something you're not certain of.
There is no trash can.
And it does not ask for confirmation, so RM is a bazooka and you can't blow your foot off.
I've deleted an entire directory of data before. Don't do that.
There's no trash can or anything. The file's
Okay, that is it for today. Tomorrow will be nothing but Unix and Linux. Your homework, Vim Tutor. Get as far as you can, but at least memorize everything in lesson 1 and maybe lesson 2. Those are the ones that can help you the most. 2, watch the videos from day 1 and day 2 on the website.
And
So
there's
there's
also
a
a
bunch of
brief.
videos. This is
Notice
the
it
first.
says homework. That's this list that she's mentioning
Yeah,
right
so if you
now.
click on homework, it will say exactly what I'm saying right now. Do your Vim Tutor, do videos from day 1, do videos from day 2. I know it's annoying to have to watch these videos because maybe you know it already, but please watch them anyway, because the only way we're going to be able to help you is if you know all the stuff in the videos and all we're doing is helping you drive. And tomorrow is a lot of driving, so make sure you watch those videos.
I just got my license.
You just got your license? We will have you on a highway by Wednesday.
Oh, that's easy.
If you still have not logged in, please stay here so we can get your computer logged in.
I can't log in.
So you are on a Windows. Okay, so wait.
Yes, so this one also, so you can move all your elements from into the regular computer. And you can move over the regular computer into the list. So it doesn't hold directions on the file.
We're getting a timeout on the bid server. It looks like the server is down.
Guys, the XML terminal may not be working right now, that one. So don't worry if we get onto the this one. We'll just work on this one later this week. You only need to say you did not get onto this one. So if you did not get onto this machine, stay around. If you didn't get onto that one, don't worry about it. We'll get you on later.
Yeah, man, it looks like it's
Maybe. Matt had that one really locked down because it's gonna cost so much to run it.
yeah, there's absolutely nothing.
Okay, don't worry about
It
that.
has exactly 0 ports. So that would indicate that it is not working.
Also, remember we have help desk, you guys need it. Anybody still need help getting onto this machine? Donnie, did you get onto this one? Okay, take your pick.
Yeah, we got it on. We got it on.
Okay, you get on that. And make sure we got everybody out.
Okay. I
Should at the very least give you something.
Now, Bobby, I have a question. I don't know about Bobby. I'm gonna add it to that stack.
It's a couple things, like you have to be on the secure network.
Okay, yeah, that's pretty normal. Rosslyn is down here in Ross too. But I know that that's not right, you know, they have so much back.
Yeah. Basically, you set up the entire presentation last year. I'm gonna still follow this today. Yeah, so
What about voting?
I'm pretty sure it's just like the violence. There's so many weird concepts.
Their supercomputer. Do you know if you guys have a monster?
No, not specifically. It's still not already. To get something going.
Not everybody uses the monitor, so it's some supercomputing I know what we're teaching is.
Yeah, because it comes with what's on it too.
That's exactly what that's for all.
How many notes?
You have it all available all the time.
So what I say. No.
There was this really cool one about some castle and
Well, I gotta go. Leave some mice and then run a flow experiment.
Oh my goodness. Have you ever used Vim?
Not really. I mostly just like browse and look stuff on
all the originals.
If you learn Vim, I know it's a pain in the butt, but this is much faster.
No way.
It takes a little while to learn. You're going to be learning somewhere to learn. But like, if I come to I know that sounds silly, but it's like. Okay.
2 and 3.
Graham, my guess is your dad's gonna be outside, so we should probably go out. We told him to pick you up at noon anyway. Let me check if he's been texting, because I didn't hear my phone. That's your mom's old laptop, so she gave you she gave you the login, you can practice computer. What'd you think of it? That's good. Yep. It's really quick to move it around.
I actually got to the last box.
So Vim is hard to learn because it's got a lot of typing commands, but once you learn it, you're super fast at moving stuff around. Moving stuff around, deleting it, rearranging it. Heck, you know what you can do in Vim that you can't do in Word? So you kind of can't, you can click the replace all button, but it's like 2 lines to replace every name in there with a different name. So it's really easy when you're like, oh, I think the name should be this instead, change the ball.
Yeah, because I'm not sure
That.
what I have was that one that you said gave us information.
Okay, so that's what's coming here.